Let's keep doing that. So I'm just going to give a brief overview of what are challenges in the field of exposomics and what I think some of the next steps are in the field. This is a symposium that we do twice a year, once in Brescia and once at Mount Sinai, and we're going to continue doing it even after the pandemic. But each year the talks are different, so please join us for future symposia. And each year we try to address different types of challenges. This year it was COVID because that seemed like the most obvious challenge. I thought also I'd give you a picture of where we would have been had the pandemic not occurred. This is the University of Brescia in Italy, which is in Lombardy, which was a province which was particularly hard hit by COVID. But the building that we were in last time actually was a renovated 500-year-old monastery that was run by the School of Economics. And hopefully at our next European symposium, that's where we'll meet. So I thought I'd give a brief introduction to what exposomics is, and it really is defined as all environment. And that includes not just chemicals, but viruses, and I think COVID has illustrated their importance, the social environment, nutrition, physical environment, air pollution, vectors, all those sorts of environmental factors that really interact with our genome. And because it's multiple types of exposures, it's also multiple types of data. So it's not actually one kind of measurement. And so one of the challenges in exposomics is the integration of multiple data types. And that's going to include things like questionnaires, whether or not what your job title is and what your job duties are actually has as part of your exposome. Even things like what's in your medical record, the fact that I've had knee surgery as part of my exposome because it's going to potentially interact with my genome. And there are other types of measures that are GIS-based, air pollution, socioeconomic status, light at night, noise, all these things can be measured, as well as photographs are probably going to become part of the exposome going forward. I think researchers in particular are interested in this. And obviously there's always going to be a role for biomarkers and how it integrates with different types of omics, such as epigenomics and transcriptomics, but also there's going to be integration with wearable devices as well. So exposomics has a lot of work to do, but I think it's probably the most important component that is yet to be studied very well, and therefore it is the area for which there's the most growth in research. So I thought I'd briefly talk about and focus on one particular challenge, and that is developmental windows and critical exposure windows. So this is an example of hemoglobin. So on the top row, you see where hemoglobin is synthesized. So in pregnancy, which is on the left part of this chart, the left part of this line here, it's in the yolk sac, then it's in the liver and the spleen. So as you go farther into pregnancy, hemoglobin synthesis actually changes organs. And the types of hemoglobin, and there's multiple types, but there's alpha hemoglobin, beta hemoglobin, which is the adult type. But there's also a fetal hemoglobin that is synthesized early in life and then gets less synthesized as time goes on. And there's some other minor hemoglobins as well. If you think about how this relates to toxicology, a prenatal exposure that inhibited the bone marrow may actually have less an effect on fetal hemoglobin if it occurs at 12 weeks of age than if it occurs at the 36 weeks postpartum. So this is part of why there are critical exposure windows, is that there are developmental factors that are changing over time, and environment is laid on top of that. And oftentimes we tend to conflate exposure windows and developmental windows. So this part of the chart is actually the developmental window. This is what the processes are. And then this bottom example of environmental exposure and how it can actually vary over time is actually that intersection between developmental windows and critical windows. And that's what we're really talking about when we talk about critical exposure windows. And it's very, very complicated. So development is sequential. Proteins and cellular processes precede the trait, and then one process can lead to another. So we tend to think of it as a very linear process. So this is a diagram that shows brain development, different types of functional domains of brain development, whether it's sensory pathways, or whether it's language, or whether it's higher cognitive functions that we sometimes call executive functions. This is actually the kind of work that I do in environmental health. And language is actually very famous as a developmental window during childhood. In language, if you talk to psychologists, the window is thought to be somewhere between 3 and 9 years of age, which is sort of this peak of this developmental process. So if that's true, then why can prenatal exposures affect language? Well, it's because in order to lay the infrastructure for that developmental window to occur, a lot of other things have to occur, oftentimes in fetal life. So you have to synthesize myelin, but first you have to have neurons differentiate, they have to migrate, they have to form different components of the brain. And we tend to think of it as linear, but it's actually not linear. It's multidimensional. All these things are happening simultaneously. And in fact, I would argue that we don't have statistics that actually allow us to study all the things that are happening simultaneously during development. Our statistics are sort of like our language. They're actually very, very linear. One leads to another, but in fact, they're all interconnected. And I think that's one of the challenges in developmental biology and research in general, is how do we start to understand this complexity of the concurrence of everything that's happening simultaneously. In environment and exposomics, many things are happening simultaneously. But we can start to get our hands around. In fact, there's some work being done statistically to look at environmental exposures and health outcomes or phenotypes. So in this graph, we're looking at air pollution in what's called a distributed lag regression model. So this black line actually represents a beta coefficient and how it changes over time. So the x-axis is weeks of gestation, and this is air pollution on the y-axis. So the beta coefficient is non-significant, and the 0 is actually the area where it's significant. And in a relatively brief window between the 10th week of gestation and the 24th week of gestation, there is statistical significance for this air pollution, which is being measured weekly. So even though many, many processes are involved, if we understand the time-varying nature of the exposure and we measure the phenotype accurately, we can actually look at whether or not statistically we can define the boundaries of these developmental windows. That's a lot of work that's being done on that in Dr. Manisha Arora's tooth biomarker, because it also has very temporally dense data, can also use distributed lag regression models to try to identify statistically where the developmental windows are in time. So what are the next steps? So exposomics really needs to be integrated into all health research because environment impacts all diseases. And I would argue that nearly all the unknown pieces of the puzzle for why we get sick are environmental. In fact, environment is the missing heritability, particularly if you define it as gene-environment interaction. And right now we're in a situation where every NIH institute funds genetics and genomics. Where we want to go is to get to the point where every NIH institute also funds environmental health and exposomics research. And in gene-environment interaction and exposomics in and of itself as a predictor of health. So there's a lot of challenges that we're going to have to overcome. And this is just another challenge. Replication is going to be very difficult. Replication is absolutely necessary for all research. However, there's a lot of considerations that have to go into replication that actually don't have to go into studies of DNA sequence. So exposure covariance matrices are going to differ across populations. They're also going to differ across time. So even if you went in the same community in 1990 as in 2020, the exposure covariance is going to be different. And it's going to differ based on age. In that community, the average 7-year-old is going to have a different covariance than the average 10-year-old. And you're going to have to factor that into replication. And it's going to differ based on geography. If you live in Detroit, which is relatively close to sea level and is very, very urban, it's going to be very different than Denver, which although it's urban, is very, very high in altitude. And it's going to differ in genetics. So genetic susceptibility is going to affect kinetics and dynamics of chemical exposures. And it's going to differ based on health status. If a population is relatively more obese, they're going to metabolize chemicals differently. Diabetes, other types of illnesses are going to affect metabolism of drugs and chemicals. And as I mentioned earlier, these effects are also driven in part by the exposure windows. You need to measure the environmental exposure at the right time. You need to measure it during the exposure window or you may miss the effect. And just as epigenomics relies on target tissue issues, those likely apply to exposomics as well and are probably a function of the toxicokinetics of the chemical. And there's other challenges. We really need to get environment into medical practice. And I would argue that precision medicine is an opportunity. And that was actually what I talked about at our last symposium. But I believe that's still true, that we need to actually get into medical practice. and medical education. Public health research is still critical, but I don't think it has to be our sole area. And we also need to illustrate how environment actually drives health disparities. It's not really driven by genetics, it's driven by differences in environment. And that's true for both research on prevention of diseases and research on treatment of diseases. And many of today's talks are going to address these issues and others, and future symposium will address them more, so that exposomics can complete the disease puzzle. That's my last slide.